Morning everybody, my name is Adrian Chavez and I'm doing the presentation for the activity on Module 5. Today I want to talk about how plant trade influences insect communities and using the Hawaiian tree Mestederos polymorpha as a model system. Even with a single plant species, different individuals can vary a lot in things like leaf nutrients, different trichomes, leaf structure, and these intraspecific differences can really change which herbivores show up and how abundant they are in a specific area. So understanding these bottom-up effects matters, yes, because insect communities often respond directly to plant quality and defenses, and classic work has shown that variation in plant phenotype can shape the entire insect assemblage. And this is not just a Hawaiian story. So across many different systems, genotype and phenotypic variation in traits such as foliar chemistry, phenology, plant site, a specific leaf area explains a large amount of the variation we see in herbivory density and diversity. So, well, to better understand how all of this is different in a single plant species, the present project is focused on a simple question. So, how does insect community composition or beta diversity vary with elevation and tree height? So, based on the patterns that we observed in the dataset and the results, I hypothesized that the elevation would explain more variation in insect community structure than the tree height. So environmental gradients often influence plant traits, which then filter insect species. So we expect insect communities to change along the elevation that the plant has. So to go over, this study was conducted across 4 mesic forest sites on the Hawaiian island. A total of 49 canopy samples were collected from 24 different Metrosideros polymorpha trees during the year 2014 to 2015. So insects were then identified to morphospecies from the different orders, especially Hemiptera, Lepidoptera, and Coleoptera. For each tree, several plant traits were measured, including trichomes, density, specific leaf area, leaf nitrogen, leaf phosphorus, and water content. These traits are known to affect the palatability that a plant can have, access to leaf surfaces, and also resource quality for herbivores. Okay, for the data analysis, what I did was to follow the R script provided in the assignment. And here we focused just on the Hemipteran order as it was provided in the R script. What I did was first to create a species by plot matrix by semen abundances and each of the Hemipteran family for every tree, and then combine it also with site variables such as height, elevation, and precipitation. After this, we calculated two different alpha diversity metrics, richness and the Shannon diversity index, using hill numbers and examine how they varied across different sites along their tree height, elevation, and precipitation gradients. To better understand how community composition was, we used a Bray-Curtis dissimilarities and ran a distance-based redundancy analysis, also known as DVRDA, to test whether elevation and tree height explain variation in insect communities. Okay, for the results, here we can see that the DVRDA showed that the elevation had the strongest influence on insect community composition, meaning that samples at higher elevation clustered together in ordination space, pulling up the direction of the elevation vector, which suggests that distinct community structure at higher altitude might happen. Also, tree height had a weaker but still detectable effect on community structure, as indicated by the arrow in the DBR-RDA. Overall, these results support the hypothesis that insect communities are structured by environmental gradients, with elevation having a more pronounced effect than tree height. This aligns with the broader idea that interspecific variation in plants, influenced also by local environmental conditions, shapes the diversity and composition of herbivory community. Thank you.